So I'm presenting a work we did in eLTER Plus for kind of developing tools for representativity analysis of the site network. And most of the technical work was done by Thomas Ohnemus, and he's not part of the workshop. But I wanted to acknowledge that before I start. And I thought I start with the research challenges of eLTER, so the main research topics we are aiming for, because this, of course, gives the framework for what the site network has to be representative. And the research challenges, there are 4 of those. It's biodiversity loss and land use change. It's all the issues we have with climate change in ecosystems. It's about air pollution, eutrophication and pollution, and sustainable management of our ecosystems. So these are the 4 research challenges, and I think most of you have at least some idea or a very detailed idea how this should look like then at a European scale. So the main characteristics of what we are developing here with eLTER is a site-based measurement network crossing all spheres, so from the anthroposphere to the geosphere in each of these sites or platforms. And of course, this system has to be representative towards the research challenges, which I showed before. So and this will be the main task of the main content of my talk now. Maybe also important is that what we develop here at the site and platform level are information clusters. So I think most of you are aware of the monitoring efforts and monitoring harmonization to achieve long-term harmonized data products from the sites is in everybody's mind. So it's the standard observations. We also try to mobilize as much as possible, and I think we will hear in one talk, we will hear about that, legacy data, so existing long-term historical data from the sites, but also from third parties, and statistical data and remote sensing products, which we then add to these information available at the site levels. So this is called information cluster. So because it populates increasingly the data knowledge of the sites. But it's also, the sites also are knowledge hubs. They should be knowledge hubs. So the main purpose of eLTER is to ease research at the site level and across sites. So it's a question of critical mass. So how many people work at the sites and how good can they mobilize cutting edge research and funding, of course. And of course, across the sites as well. And one of the means to do that is co-location. So adding many measurements, some might come from other research infrastructures than eLTER, to the sites. So to increase the measurement potential, or say the scientific potential of the sites, and also open access to the sites and the data, of course. This is one of the pillars of eLTER. So starting with that, I mean, eLTER is based on a scientific theory, which at the site level is the pulse press dynamic system, which Collins et al. Published in 2011, where we as much as possible try to reflect the pressures at an ecosystem level, at the site level, taking into account long-term trends, but also presses like extreme events, for example, and also taking into account the social economic drivers at the site and platform level. But for this talk, more important is how we intend to upscale. So and there the scientific theory is the microsystems ecology theory. Heffernan and et al. Published that in 2014, more than 10 years ago. But there has been a lot of development in that, how we are able to upscale very complex processes, where from the local to the macro scale, there are cross-scale interactions and cross-scale emergences, which we don't see at the site level, but we have to take them into account. And there's obviously also a lot of interactions going on the global scale, like climate change, for example, La Nina phenomenons and these kind of things. So, so there are the two theoretical frameworks. One works more on the local level, and the other one deals with how we upscale and downscale from all the site data and all the site knowledge we generate. So we worked towards analyzing sampling bias, representativity, and transferability. So when you take an ideal scenario, the conditions in a in situ facility, a network of a research infrastructure could be to precisely reflect, of course, the target region. And the target region in our case is Europe, let's say, at least all the member states which are joining eLTER. But in reality, this is not the case. So there's always some kind of sampling bias. I mean, just think of how eLTER sites developed. So there is a lot of like history in how these sites developed, and there was no never a top-down network design in place to locate all the sites and platforms. So there's of course a bias. And we use two approaches. So it's the representativity approach, which compares the value distributions covered by the in situ facilities with the value distribution of the target region, and transferability investigates more the similarity in the conditions of the in situ facilities and the target region. So these are two approaches, standard approaches for addressing sampling bias, and I will go through it. Through these studies and results we achieved using quite complex spatial analysis. But I start with the structure of eLTER, just that you are aware of that the distributed physical network, they are kind of independent from the central structures of the upcoming research infrastructure, ERIC. And there is a lot of political support and funding support necessary to keep the sites running, or even to create new sites. So there is quite some support. You see all these like blue-colored countries where there is political support, and a lot of scientific institutions support eLTER. So it can capitalize on a lot of sites, so more than 500 sites, mostly from eLTER Europe, actually, and more than 50 LTSCR platforms. So this is kind of the pool from which all this endeavor towards the research infrastructure started. Some years ago, there was a category screening going on. So all the national coordinators were asked to bring in their ambitions, let's say. So how these sites, which is the pool with 500 sites, what their status is currently, and how they are going to be implemented when the research infrastructure is starting. And I think most of you should be aware of the categories, or the different categories very well defined in the eLTER. Let's say category 1 is a very ambitious site with a lot of measurements, complex methods. Category 2 is more like the basic category where all the long-term data is measured with a defined protocol. And category 3 is kind of an associated site which tries as good as possible to work towards the standards developed in eLTER. So the screening resulted in this so a lot of like ambitions from the current to the future situation, a lot more category 1 and 2 sites than currently, and all in all we came up with more than 200 category 1 or 2 sites across Europe. So this is the most likely, from what we know now, the most likely network we will have when eLTER is launched, when the eLTER ERIC is launched, and then during the next years, let's say, when these measurements are implemented. So this was also the basic for our analysis. So this is always the current situation is 224 sites, and the question was how these sites represent what we're asking for. So there was quite some time ago. There was a lot of there was the basic say analytical development, how we deal with this kind of bias calculations, and this was the first study done already in 23 and published in 24, and there the main question was which spatial gaps we have. So where might be priority regions for additional sites, so which areas we do not cover well with regard to current conditions. So the conditions, they involved anthropogenic biomes. Here are all the citations for that: biogeoclimatic zones, bioclimatic zones, economic density, which was the GDP per capita, population density, land cover, landforms, and biogeographical regions. So these 7 layers were used to define the current situation. And by doing, and bear with me, please, I don't go in the detailed methods. I think Giorgio distributed all the papers related to that. There's just not enough time to go in all these details. More focus on the results, actually. So there were, when doing the representativity analysis, we found that the bias is mostly there because we have distinct gaps, which is the Iberian Peninsula, Eastern Europe, and North and most Europe, where we do not have enough sites which cover the 7 layers of the current environmental and socioeconomic situation in Europe. So these are, so the reddish, the redder the colors are, the higher is the priority for additional sites in these regions. So, and also there was also we also at this time analyzed how the different ecosystem types are represented, and there's of course an over and under representation and an over representation. Sorry, an under representation, which is more important, like in context of network design or achieving a better network design, is that the underrepresented sites were mostly grasslands, even forests, because there's also forest area in Europe. So to cover that unbiased, you need a lot of sites there, and vegetated man-made habitats, so agricultural areas, more or less. And there was, of course, also some overrepresentation, but this is, I think, in this context, not so important. Okay, so this was the current situation. So with the 224 sites, we expect to have in eLTER, in the eLTER RI, there are very distinct gaps, and we can communicate these gaps to policymakers or like funders at the national scale, how they might be able to fill these gaps. But then we thought, okay, so this is nice. But the world is changing, so there's very rapid climate change, also socioeconomic change and land use change going on in Europe. And how would these sites, how would the network design look like in these very likely futures? So is the network fit for the future as well? So this was the question in a second work we did, and we used ensemble approach. So a couple of climate scenarios from RCP 4.5 and RCP 8.5 scenarios, and from these climate scenario data, we developed indicators for climate change, land use change, which we think are affecting ecosystems significantly in the future. And so we focused on warming, so which is we call this biotemperature pressure, which is kind of the speed of warming. And these the scales, this is the indicators are scaled towards plant function types. I don't want to go into detail there, but it mostly assumes that this scale reflects a change in plant function types. So there was a warming indicator, there was a drying wetting indicator focusing on precipitation changes, a land use change also linked to these scenarios. And what's not shown here is we used a drought index to reflect seasonality changes, so which we expect. So, for example, a shift in towards drier summer or wetter winters or whatever. So these maps, they show whenever you have these crossed shapes, then there's no significant changes going on there. So this was a spatial analysis done by Thomas just to get these metrics right and then compared it to our site network. So maybe an easier take on how this looks like was a hotspot analysis where for each of these pressures you look at the distribution across Europe and you see where the main change is going on. So with regard to warming, this is mostly in the high elevation areas and in the Mediterranean and northern Sweden and Finland. It's mostly precipitation changes which are going on there, increases and decreases. With regard to drought seasonality, it's mostly in Central Europe, for example. And then you see also a scattered distribution of very strong land use changes across Europe, expressed in socioeconomic scenarios. So doing the same analysis as before, there is of course then a picture, these results in maps of under and over representation with regard to each of these scenarios, and then to the main result on the right side here, showing again the representation, so the priority areas where additional sites are recommended. And when you remember the map before, it looks quite similar actually. So we again have the gaps up here in Insula, it's smaller in the very southernmost Mediterranean area, some scattered gaps in Eastern Europe and northernmost Europe. So the gaps are the resulting gaps with regard to the future fitness of the site network are very similar. So what was the conclusion? So when we finished that, it was that the highest priority maybe is the closing of the major gaps which I just mentioned. There was also, the analysis also showed us that they have to be treated independently. So it doesn't help if you close just one gap and then the bias is improving that much. You have to really independently do that because the conditions are very different in the different gaps. What we also found is that when you close the gaps for the current conditions, you mostly also cover the future fitness. So you also improve the future fitness, which was a good result actually. It would be very difficult if a totally different picture came out. So it's very similar. So individual targeting of the gaps and also targeting specific features helps a lot in bias improvement. So, for example, sites in agricultural areas of low income economic density. So this is definitely a target for additional sites and areas that will experience substantial pressure due to land use change and precipitation changes. So this is also one of the possible targets in the future. So with that, we didn't stop there because we asked how. So how can we improve? So really practically, how can we reduce this bias? And couple of years ago, the co-location like concept we also published in our work package, mostly done by Martin Futter, who wrote a very nice paper in Ambio actually on this topic, where the main idea is that we have eLTER and most of the sites are in eLTER Europe. There are also a lot of additional research infrastructure and monitoring networks which we can leverage for eLTER, or let's say the national coordinators can leverage for eLTER. So adding these sites into the eLTER network, which is cheaper than putting new sites on a place, of course, and it adds a lot of capacity at the site level too. So this is an example of a site we run actually. So it's Zilberboden in Austria. And there we, for example, already co-locate a couple of monitoring and research networks, for example, ICP integrated monitoring. This was the basic infrastructure start for this kind of long-term monitoring site. We also are in eLTER, of course, newly also in ICP forests. It's an EMAP site for high quality air pollution measurement. It's a life plan site for biodiversity monitoring, and it's a FluxNet site, so measuring carbon balances with eddy covariance stars. So this kind of co-location and linkage to many different networks and monitoring networks research infrastructure helps a lot in improving not only the condition at the site, it can also be a possibility to, in a cheaper way, improve the sampling bias at the network level. So this was the idea. So we collected all the site networks, the locations from a couple of say logical partners, because there's some cooperation already going on. They're doing similar things. There's long-term data and long-term operation of the sites in place and so on. So it was a couple of research infrastructure. So ACTRIS, it's also a European research infrastructure for atmospheric research. FluxNet and ICOS, the most famous carbon site networks measuring biogeogenic fluxes of carbon and other greenhouse gases. Then there is an Arctic alpine ecosystem research network, INTERACT, in place, and also very famously the monitoring network, which was established for air pollution effect control in Europe already in the 80s. So probably the oldest and longest historically time trends we have for ecosystems in Europe. So it's ICP forests, a lot of plots of ICP forests. There are already, I think, 100 ICP forest sites in eLTER, but not all of them. So there's a lot of additional sites actually. It's the level 2 network for from ICP forests. Then ICP waters and ICP integrated monitoring. The slide I showed before from Zilberboden is an integrated monitoring site, so a little less than 50 sites across Europe. So we use these sites with the rationale that these are the most logical sites to use and add to eLTER. To the 200 sites, and these were more than 800 sites, 800 candidate sites actually, which are available, which are not yet in eLTER. So we checked that these 800 sites, they are not yet in eLTER, but they could be part of that. So for the first step, we implemented a randomized iterative process where we randomly choose a site, and then we calculate the improvement in the representativity and improvement in the transferability, and we do this a couple of times, and then we select this site and add it to the existing 224 sites, and then we calculate the bias reduction. So this is the result here when we're selecting 50 sites. So why only 50 sites? So it's just reasonable. With regard to just when you think about funding and so on, it's just reasonable. We probably not get 200, and maybe we get 500, but more reasonable are like 50 more sites. That would be already really nice to have that have more than add 50 sites to the 224 sites we already have in eLTER. So this was an assumption. So, and then we calculated the bias reduction, what you see here for the different facets, let's say, so for the current conditions, but also for the future conditions. So taking these two RCP scenarios for the future fitness with regard to climate change and land use changes. So what I see here is like there is, of course, a bias reduction. So the bias is decreasing. So the whole network design is, of course, improving. I mean, this is not a surprise, of course. I mean, this was quite logical that this is going to happen. But what was surprising that you don't get the total bias unbiased sample. So there's still some bias remaining in the current conditions. It's best in the RCP45 scenario, and in the RCP85 there's also a bias remaining. At least 60% bias is remaining. So the conclusion from that is that co-location is good. I mean, it's obviously a practical way to improve the network design, but it's only half of the story. But anyway, what we were able to do is we were able to rank more or less 200 potential co-location sites, which is a good, let's say, tool for also the national networks, because with this ranking, they exactly know which sites are the best sites to include in eLTER from the best perspective of network design. It's also an argument, let's say, for like funding. So to say that with this site, I'm really improving the network design of eLTER, and as an additional argument, not only which goes beyond like the argument that at the national scale you want more eLTER sites. So then there are many potential candidate sites in the gap regions. So the gap regions can be improved. So this is shown here in the graph. So in the upper panel, there are the situation without co-location. So these already classical gaps here, which are shaded in black, showing these Iberian Peninsula gap, Eastern Europe, northernmost Europe, where you see these bigger shades here, where we have the most severe problems actually with regard to network design. And this is improving. So with the co-location, these gaps become smaller. They don't diminish totally, but they at least get smaller. And in Eastern Europe, it looks pretty good actually. So they are almost gone. So there are few candidate facilities. With the gap remaining, for example, in the Iberian Peninsula, and southernmost Mediterranean areas, the take home is just there's not enough candidate facilities. And with even after the addition of the 50 co-location sites, there is bias there persists some bias, and of course these areas which are still reddish are the focal areas where the new sites probably have to be developed to reduce the to reduce bias even more. So what we achieved here, and what's the work ahead of us is the following. From the beginning on, eLTER was very good in like leveraging sites from many existing research and monitoring networks. So I mean, many of those working at the national scale, they know that the topic eLTER is everywhere, and many of the site managers try to get into the network because there is, of course, science and funding benefits from that. So also with regard to co-location, there's a lot of collaboration going on with ICOS. There's a very long-term collaboration with ICOS going on, especially with the ICOS ecosystem sites, of course. And also a very tight cooperation exists with the air pollution monitoring effect network, all these ICPs. And this is also already reflected in the picture that there are many eLTER sites in parallel, other like ICP forest sites, integrated monitoring sites, and so on. So this is good and this works. This has to be tightened, of course, but there was this is an achievement, I would say. Another achievement is also that with regard to the harmonization of the protocols from the beginning on, it was the strategy to use existing protocols to ease co-location, let's say, at the site level. So because then you, of course, don't have to measure things twice if you report your data to different networks, you only have to measure it once, and it goes to for different purposes to eLTER and your maybe traditional monitoring network as well. So this was also, I think, an achievement which gives us leverage to improve the network design. There are still we have priority sites, so we know the priority sites, so we can really pinpoint single sites where we know these are the target sites. This was also communicated to the national coordinators and so on. So at the national level, you know, I mean, the further development of eLTER site and platform network is mostly run by the national coordinators. So because they have to get the funding and bring in the sites and so on and so on. So this is where this information is most useful, of course. What we also think is that guidelines, they don't exist yet. I mean, maybe some training or so would be very, very useful into how at the site or platform level co-location is done. So how do you technically do that? So best practices exist, and it would be nice to have some like guideline best practices training on that. Nevertheless, there are new sites necessary if you really want a totally unbiased network, which is probably a vision, but it's just a fact. We defined the priority regions. So this is if anywhere funding comes up for additional sites. So these are the regions where, and the features we can pinpoint where additional sites are necessary. So with that, I think I'm finished, and I leave it here, and I'm of course open for questions.
Thanks, Thomas, and for the very detailed presentation, and really on the point also for the workshop objective. So I open the floor for question. You can raise your hand, or also if you write the question in the chat, I will read to Thomas. So Ricardo.
Yeah, thank you. Good morning, and thanks, Thomas, for this very, very interesting outcomes from your analysis. I just have a very quick question. Is as the main final objective is to have upscaling of the in situ collected data, I wonder why we didn't have into account data provided by remote sensing, because we've been struggling in the work package 4 and especially in the task 4.4 to propose a pilot exercise for several eLTER sites in order to enlarge upsample, we call it this way, to make the complementary data provided by remote sensing sources such as Copernicus, not only reflectance, but also products that could be eventually validated, and we have demonstrated so with the data collected at in situ level, and then expanded, enlarged, as I said, to the complete, very, very larger regions, let's say. So my question is this: how could we eventually reduce this bias by including in this upscaling exercise to have a pan-European information system in order to enhance the data we collect as a research infrastructure? Thank you.
Mm-hmm. Okay. So when I get it right, you don't mean that in the analysis we should have used remote sensing products, but it's more like providing information at the European scale with which we can work in addition to the site data, isn't it?
Well, actually, the point is that if we find a way to fulfill the gaps at, let's say, local and regional scales using remote sensing in a way that we can eventually validate some of the products as they are, become standard observations straight from the remote sensing data from the Earth observation, we could eventually provide new data to carry out the analysis that you've been doing, like trying to understand, including this ancillary data could be enhancing or reducing the bias what we're looking for. So
yeah, no, I couldn't agree more. I mean, the what we did so far was I see it only as a kind of a start. So what I understand is that these analysis they are there, and this is a workflow which is there and can be extended. So with exactly these kind of questions, so how can we add in remote sensing products and also modeling products? So I mean, I think you are aware that we also strive for. I think the next talk will cover that, or by Christian, he will cover that a bit. That also a not only remote sensing product, but ecosystem system science model products are going to be developed for the network, which is also then filling not only the gaps in the data from the measurements at the sites, but all the areas in between, of course. So like the same as the remote sensing products. And so, I mean, the tools are there, and I'm sure this is not the end of the story. And I actually hope that we are going to add both the remote sensing and the modeling products to improve bias.
Thank you, Thomas.
Thank you, Ricardo. Thank you, Thomas. There is a question from James Weldon from SLU in the chat. So if the list of identified priority sites is already published somewhere.
Sorry, I didn't get it fully. All the three projects or all the three parts?
If the list of identified priority sites is
No.
published. Okay.
No, but it's in review, so it will be published soon. And but in the paper you don't find, of course, this priority list for the site. So this is in the head office. So I think it's available via the head office.
Okay, and I have two questions actually, as I don't see any other hand raising. And the question are the resolution, let's say, of the map and of the analysis. So which is let's say the spatial resolution, and if in your last analysis of let's say possible priority site, have you included the current, let's say, category 3 sites of eLTER for possible improvement to category 2 and 1, or these are already in the first analysis?
The category 3 set, this was a little bit of an issue actually, because with the category screening, the category 3 sets were not covered very well. So the labeling from the national coordinators with regard to category 3 sets, I think, was very arbitrary. So some did it, some did it not. So this was not a very useful data set. So we stick to the category 1 and 2 sites, and then added all potential sites from the other networks, which I think are a better representation of category 3 sites anyway. So with regard to resolution, so this was very different in this different analysis. So whatever we had, we used, let's say, you know, I mean, this classical climate scenario data and the others. I'm not even aware actually from the first study. I wasn't involved so much there. I think there was also very different resolutions of this data.
Okay, thanks. I don't see any other hand raised. So thank you, Thomas. And actually, the material will be available, and also the presentation, and for also work in the next coming days. And so I invite for the second presentation Sinclair and Peter Haase. I don't know who is going to present.
Oh, Peter, you need to rejoin. Peter will be right back. This happens sometimes.
Okay.
It usually only takes him like 10 seconds.
Okay.
It's a long 10 seconds.
Okay, but we refresh our mind for the coming presentation.
So I'm back. Hopefully you can hear me now.
Yes, better.
Should I start or?
Yeah, if you are ready, you can start.
Okay. So I hope you can see my full screen now.
Yes,
so,
perfect.
Okay, great. So this presentation will be given by me and my colleague James Sinclair, and this is about multidecadal trends and drivers of biodiversity change, linking eLTER to continental scale biomonitoring. So our presentation comprises two parts. The first part, presented by me, will be on a very brief history and also providing some highlight examples, addressing aspects like how to scale up data from eLTER sites to the continental scale, and also how to link eLTER to the broader scientific community. As an example, we are using data calls and also how eLTER could actually be perceived as an honest broker. And finally, we will provide examples on how to jointly explore data and write scientific manuscripts. And the second part that will be presented by my colleague James Sinclair will be on biodiversity data, excuse me, structure, harmonization, and analyses, specifically providing an example how to link temporal community change to ecological quality, and also addressing some common problems that you may encounter when you're doing time series analyses, and also providing some potential solutions to these issues. So everything started with an idea, and the idea was actually to make use of the eLTER treasure, which are our long-term data, of course. And the aspect we were focusing on was to analyze biodiversity trends and drivers across the whole of Europe. And for this, we launched a biodiversity data call in 2018. And two out of several criteria for these data were that we were asking for a minimum length of the time series of 15 years. And within these time series, there must be a minimum of 10 sampling years. And these criteria largely exceed common standards because some of you may know, specifically when you're looking at biodiversity time series studies, they often focus on snapshot data, so you may only have information on your first year and on your last year, and then you're simply calculating a linear trend, which might not be that much reliable. The call was quite successful, so we were able to collate 161 long-term biological time series from eLTER sites, covering 15 to 91 years, and including information on around 6,200 marine, freshwater, and terrestrial taxa. We tested whether local long-term biodiversity trends are consistent among bioregions, realms, and taxonomic groups, and also whether changes in biodiversity correlate with regional climate and site naturalness. So our statistical approach comprised 4 different steps. In the first step, we calculated for each time series monotonic trends over the study period, addressing abundance, taxonomic richness, Simpson diversity, and temporal turnover. In the second step, we applied a meta-analytical approach to calculate overall trends and also identify patterns among biogeoregions, realms, and taxonomic groups. In our third step, we conducted a sensitivity analysis, which is really important when it comes to these kind of data, because this accounts for the unbalanced distribution of our time series across biogeoregions, realms, and taxonomic groups. And finally, in the fourth steps, we applied meta-analysis mixed models to address to which extent climatic trends and site naturalness may have an influence on our biodiversity trends. So when it comes to the results, so overall, we identified significant increasing trends in richness, Simpson diversity, and temporal turnover, but no trend in abundance. So to address our second question, if there might be any differences, we were focusing on the biogeoregions that you can see here on the left-hand side, that the different biogeoregions in Europe are provided in different colors. Then we were focusing on the different realms, so specifically the marine realm, the freshwater realm, and the terrestrial realm. And we included in our analyses 8 different taxonomic groups, ranging from terrestrial plants over terrestrial invertebrates, including aquatic invertebrates and fish as some examples. So when it comes to the bioregions, that you can see here in the upper left corner, addressing the predictor abundance, that only in 3 bioregions we observed significant trends. So indicated in green, there is an increasing trend in abundance in the North Sea area, while there is declining trends in UK and also along the western coast of Europe. With regard to richness, we also have found only a few significant increasing trends in richness limited to the northern part of Europe. Same with diversity. And there was a more consistent trend in turnover. Now we are at the lower right corner, where you have a lot of significant increasing turnover values and only a decline in turnover in the North Sea area. When it comes to the realms, you can see there are no differences among the 3 realms, terrestrial, freshwater, and marine, when it comes to abundance. This is different for richness, where there were increases in richness observed in marine and freshwater realm. For diversity, it is limited to marine realm, so the marine realm, and for turnover to the terrestrial realm. Also, with regard to the different taxonomic groups, you can see the responses were mostly non-significant, with a few exceptions. So there are declining abundance trends in the terrestrial butterflies, and also in when it comes to richness in birds and in freshwater invertebrates. Similar, the pattern in diversity, and when it comes to turnover, we only found significant increases in terrestrial plants. So to sum this up, overall we found significant increasing trends in richness, diversity, and temporal turnover, but not in abundance. However, we also showed that the trends largely differ among biogeoregions, realms, and taxonomic groups, and increases in temperature and site naturalness were important drivers of the increases in richness and abundance. So what has started with an idea and the data call ended in a nice joint publication in Nature Communications in 2020. And you may recognize several names in this long list of co-authors. Encouraged by this success, we launched in 2020 a second data call with some significant differences, because this time we were focusing on river invertebrates, so just one ecosystem and one taxonomic group. And we were expanding the call beyond eLTER, so we were not only asking our eLTER colleagues to submit data, but everyone that is dealing with freshwater time series in Europe. And actually, we were hoping that we could somehow sell eLTER as an honest broker, because when it comes to data calls, then people who are addressed may often ask themselves, how reliable are the people behind the call? Can I really send my treasure, my long-term data to these people? And here we were hoping that eLTER is perceived as an honest broker, and we're hoping to get a lot of feedback. And as it looks, this really paid off, because we were able to mobilize 1,816 river invertebrate time series, which is compared to the 161 time series that I have presented in the first study, more than 10 times more. And if you're then considering that this is really limited just to river invertebrates, this was really an amazing success. The data and the expertise has been provided by 96 co-authors and covering 22 different European countries. Okay, so sorry. We addressed two main questions. So the first was, how have freshwater invertebrate communities changed over the past 5 decades in European streams and rivers? And the second question was, what environmental factors have driven these changes? So here I only provide one example, and the example is taxon richness, so the number of species observed, and here specifically how the number of species has changed within each of the time series. And you can see here indicated in red the amount of time series and also the extent of annual change in taxon richness. And the red color indicates a decline. So there were quite some sites where we observed a decline in taxon richness to a small extent, and only a few to a larger extent. But for the majority of sites, and this is indicated in blue, you can see a clear increase, annual increase in taxon richness over the observation period. The overall average across all sites is an annual increase by 0.73% in year, and this is adding up to almost 50% overall increase in taxon richness across Europe over these more than 5 decades. So as an eLTER person, you are actually maybe a little bit more interested in how do these trajectories look like. So these are just averages, but this could actually result from a continuous increase in richness, but it could also be include some fluctuations. So starting with a sharp increase at the beginning and then some decline, or the other way around. And this caused some headache on our end, but then we came up with a potential solution, and we applied a moving window approach, a 10-year moving window approach. I'm leaving out the details here. But what you can see in this graph is that on this axis, it's again taxon richness, so the percentage change per year. And you can see that in the 1990s, across all sites, the average increase in taxon richness was around 1%. It's a little bit fluctuating. And this is still the same in the 2000s until 2010. And then there is a sharp decline in taxon richness, which is not increasing anymore. So this means we have observed 1% mean annual increase in the 1990s and the 2000s, and we interpret this that past conservation actions have paid off, but these increases leveled off after 2010, and this is due to remaining and new stressors. To address our second question regarding potential drivers of these changes, we were looking at temperature increases, and here you are actually, here we are showing how increases in temperatures or temperature trends are related to taxon richness trends, and indicated by the red color. You will see that these are contradictory trends, so meaning that if you have an increase in temperature, then you have a decline in taxon richness and the other way around. Meaning also if you have an increase in dams, you will also observe a decline in richness. If you have an increase in urban area, again, this leads to a decline in taxon richness, and the same for an increase in cropland coverage. To sum this up, we could clearly show that previous conservation measures paid off. So specifically the continental scale implementation of wastewater treatment plants during the past decades. However, still 60% of all European rivers have not achieved a good ecological status, requiring additional measures that need to include a reduction of micropollutants, pesticides, and fertilizers, but also an improvement in the existing wastewater treatment plants and more restoration and protection. So, and this endeavor, so to say, ended in a Nature study, now with even more co-authors listed here, several from eLTER and many more from non-eLTER, from the non-eLTER community. Already during the writing process of the Nature study, my mistake. So with regard to the linkages of this example to eLTER, we could clearly show that a high frequency of biodiversity monitoring is really decisive, and the same is true for driver variables, so including physicochemical data. And also, in some cases, we observed that the biotic and the abiotic monitoring did not happen at the exact same site, which is, of course, everything but not idle. And here, actually, eLTER's whole system approach covers these requirements, and this is why we are looking very much forward if eLTER is full running and the whole system approach is applied. So already during the writing process of the Nature study, we already saw that this is actually a great experience, and we launched already in 2022 our third call. And we expanded this call to other freshwater taxonomic groups beyond invertebrates, including now macrophytes, fish, diatoms, and algae. And we also expanded from running waters towards ponds and lakes. And a big surprise was that we now received around 35,000 time series from 30 European countries. So this is compared to the 1,800 in the Nature study, 20 times more. And we also doubled the number of experts and data providers to now more than 180 persons. And this is a still growing network. Just for a comparison, some of you may know that Pinsky et al. Published a nice study in 2025 in Nature, and there they were including around 42,000 time series from marine, terrestrial, and freshwater assemblages across the whole world. So we have almost as many time series from Europe and only for freshwater compared to this global study, including everything. A big difference is still that we have a very high resolution in our data set, and that we made a lot of effort to harmonize our data. And this is something that will be picked up in James' talk. So what comes next? So this incredible data set combined with the Frontiers Planet Prize that was awarded to our nature study in 2024 with 1 million Swiss francs, which is about a little more than 1 million euros. And considering that the prize money needs to be spent on a similar topic, we decided to found a synthesis center on freshwater biodiversity change in Europe. And this synthesis center comprises 6 main tasks. So the first task is centered around data, collating data, cleaning data, storing data. And for that, we already have developed templates and R scripts to automate these processes. Of course, in the center are state-of-the-art analyses, so calculating biodiversity trends, possible drivers, baselines, and so on and so forth, because this amazing data set allows to test a broad variety of hypotheses and also answering applied and basic research questions. This part is additionally supported by a current Biodiversity Plus project called De-Stress. Task number 3 is to implement the Forum for International Experts. As mentioned before, we are in touch and collaborating with more than 180 different European experts, and we are actually using their knowledge and their expertise with regard to data, with regard to analyses, and also with regard to the writing of manuscripts. We are now about to implement a platform for transdisciplinary partnerships, so also bring on board authorities, NGOs, and industry and business. And specifically, this part is additionally supported by some budget from the Airport Frankfurt Foundation. So the 5th task are to develop virtual research environments. And these research environments actually allow specifically stakeholders to analyze complex scientific data by themselves without having detailed expertise in statistical analyses. And of course, for such projects, it's always important to have a module dealing with communication and outreach, to write the scientific papers, to write research proposals, policy briefs, press releases, and so on and so forth. And for this, we also have some support from the current Horizon Europe project, BMD. So, and this is a picture not from a tropical rainforest. This is actually a floodplain forest in the Rhine-Main metropolitan area close to Frankfurt. And you can see here my colleagues taking some benthic invertebrate samples from a German stream. And by this, I'm saying thank you and handing over to my colleague, James Sinclair.
Thanks, Peter. Just a question. We leave question and answer at the end of James' presentation also for yours?
I would suggest so, yeah.
Okay. Okay. So for the question we will do after the James Sinclair follow-up presentation. Thanks.
All right. Hi. Let me get my screen shared here real quick. Hopefully everybody can hear me and there's not any real problems with that. Can we see the talk okay? We're good? Visible
Perfect.
on screen?
Yeah. We hear
Perfect.
you and we see the talk, the presentation.
All right. So hi, everybody. I'm James. I'm here to talk to you a little bit more today about some of the work Peter and I have done looking at changes in the biodiversity of European river invertebrates. So of course, Peter showed you some of these results earlier where they were looking into changes in a variety of different biodiversity metrics through time. And so the example here is taxon richness. Just give me 1 second, my laser pointer. So of course, generally increasing taxon richness in European river invertebrates over the last, let's say 30 years. So, some of the questions that Peter and myself and others had after completing this project was, well, do these changes in biodiversity really mean what they think they mean? Do they really indicate improving ecosystem health? And then, this kind of stalling of that process later on in the 2010s. And this kind of links into broader questions to biodiversity research and global biodiversity research in general. For example, this is a study from 2014. Here on the y-axis, you have log transformed species richness, and here on the x-axis through time. And so, they're often looking at how biodiversity is changing globally through time. And a very frequent result is that the overall trend, which is here in black, is flat. There's no overall change. So all of these communities are kind of all doing different things. Some are increasing, some are decreasing, some are experiencing no changes whatsoever. Now, this doesn't really make a lot of sense because we're supposed to be in the 6th mass extinction crisis. And so we're supposed to be losing taxa kind of generally everywhere. And so why can't we detect these losses when we look at local scale biodiversity trends? Well, one of the potential explanations that we had thought about is, well, maybe it's just this common issue of context dependence in ecology, that communities do different things in different places. So they could all be experiencing kind of similar degradation and climate change, but it doesn't mean they're all going to be losing biodiversity, losing abundance, losing richness, or gaining abundance or gaining richness or whatever. And this, you can kind of imagine that when you're accumulating these data, either from eLTER sites or from across the globe or whatever, that even if all communities are responding to the same environmental changes, it could create this heterogeneous mixture of a variety of different responses. And so really, does biodiversity tell us anything about anthropogenic impacts? And again, this links into Peter's talk of, okay, we see increases in European river biodiversity, but does that really mean that things are getting better? Or could this just be kind of, communities responding in different ways, and they all are maybe gaining taxa for a variety of different reasons that don't really have to do with things getting better? And so Peter really wanted to attack this question of, okay, do the things we see happening, do they mean what we think they mean? So we need a couple different things to examine those kinds of questions. First, we need to know how different community metrics, so things like abundance and richness and Shannon diversity and evenness and turnover, how are they changing in relation to anthropogenic impacts? And we need that information for a variety of different communities. Now, this community data is kind of what the eLTER research network is sort of ideally designed for. You need a lot of community data, and you need it across a fairly broad spatial scale, and you need it for a lot of different communities. So we had that data, and additionally, we had the data from a variety of other contacts. You can get them from publicly available databases. You can get them from researchers you're in contact with. You send them an email and say, hey, can you send us this data? Where we kind of ran into more of an issue is this part of the need, which was we need data on changes in impact. Now, a common problem you run into is requiring matching environmental data when you want to look at changes in biodiversity and try and understand why it's happening. But you don't always have matching environmental data. It's a common issue that a lot of people run into. But we actually managed to find a solution for it, which is in what is called ecological quality, which is a community-based measure of impact. Basically, all you have to do is you have an observed community, and then you have a reference community from the same region. The reference community is supposed to kind of ideally reflect what the community would look like in, if anthropogenic impacts were fairly low. And so ecological quality is the difference between the two. If you have low similarity in your observed community to the reference community, so these would be, values kind of around here, closer to 0, that indicates anthropogenic impacts are pretty high. If you have values close to the reference community, so it's pretty similar, values around 1, that indicates the observed community is pretty good. Anthropogenic impacts are probably pretty low. So we sent an email out to everybody who provided us data and said, hey, can you please calculate these ecological quality values for us, because you need expertise in each country. The metrics are designed to compare to reference systems in each region, and you need some expertise to kind of navigate that process. And so we had a lot of people say, yeah, sure. And so for the 1,816 sites Peter had mentioned, we received ecological quality data for 1,365. So we got a good chunk of the data set with, this community-based information on anthropogenic impacts. All right, so that kind of set up Our plan is that we're going to compare changes in a variety of different community metrics, and we're going to relate those changes to changes in impact, in our case, changes in ecological quality. And what we're really trying to find out is, okay, if ecological quality changes, let's say it gets worse, so impacts are increasing through time, do we also see matching changes in these community metrics? Do we, for example, see matching declines in richness or abundance or Shannon diversity or whatever? And do we see that consistently happening across all of these different sites? If we do, it would indicate that we can use community biodiversity information to make inferences about changes in anthropogenic impacts. If we see things go down, probably suggest things are getting better. If we see things worse, if we see things go up, probably suggest things are getting better. Or do we not see this? Is everything so variable that there's no clear pattern across communities? All right, so the next part of the question is then we have our plan, but we actually have to quantify biodiversity change through time and ecological quality change through time. There's a variety of problems that people encountering eLTER biodiversity data and a variety of other types of biodiversity time series data are going to run into. We don't have time to delve into every single problem you're going to encounter, but these are some of the main issues that we've found in our process of dealing with these kinds of data. The first set of issues are that you're going to run into problems of comparing years within a time series to each other. And so some very obvious issues with this are, of course, you might need to transform your data. That's a very obvious one that everybody's probably encountered at some point. And that's commonly an issue with metrics like abundance, where you can have these really high value swings in individual years. So hopefully that problem is fairly well dealt with by some kind of data transformation. The log transformation is always very common, of course, but there's other options for dealing with that issue. You may need to account for how changes in abundance may then affect diversity. So you can require in years with higher abundance, more sampling effort to correctly collect the full diversity of that community. These kinds of issues can be dealt with by switching. Instead of using, for example, like richness measures, you might use rarefied richness or coverage-based richness. These can deal with these problems of changes in abundance affecting your need for higher sampling effort or lower sampling effort. You can also, of course, run into issues where the normal distribution isn't really providing you a suitable temporal model. You might run into this issue with anything like count data, for example, with richness that can require non-normal distributions like the Poisson or the negative binomial distribution to really get an accurate model. Oftentimes, though, it's usually fine. The linear model is pretty powerful most of the time. Of course, run into problems of temporal autocorrelation between successive years within a time series. So for instance, you may have a point that is, high in one year and then high in the next following year, and that's because the previous year was high. So the points are not independent from one another. They are related to one another. So you have to control for that problem. Now that problem is maybe not so bad depending on the structure of the time series, right? You can have time series that have more gaps between the years, like this one perhaps. So it reduces this influence of temporal autocorrelation. But if you have a lot more data or more high resolution or a longer time series, then this problem can become a real issue that you need to deal with to get accurate slopes. And there's a variety of ways of doing that, which I'll mention in a few minutes. Lastly, I think this is the last point, you can also have some years that exert undue influence on the slopes you extract from your time series. And there can be a variety of reasons why that happened. Maybe one year was just, the conditions were really weird or something happened at the site that caused a problem. There's several different ways of testing for that problem. One of the main ways would simply be to remove particular years from your data set and see if it has a strong influence on your slopes. If they remain mostly the same, then you're probably fine. But if you see a dramatic difference when, for example, we removed 2017, everything's completely different, then you may want to take a closer look to see what's going on there. You can do it through modeling. If you have individual years in your model, you're not just extracting slopes. You could include something like a mixed effect, a random effect for year, which will help control for maybe individual years are very different compared to others. And obviously, there's other options for dealing with that, but that should be fine. You can also use weighting, so you can downweight the influence of particular points in your time series. And so there's a lot of options for dealing with that problem. All right. In addition to problems within a time series, there are also, of course, going to be problems when comparing changes among different time series. Of course, there's going to be spatial autocorrelation potentially. So you may have sites that are very close together to one another that are similar simply because they're close, and sites far apart from one another that are different simply because they're far apart. So you may need to test for and control for spatial autocorrelation, which I'll talk about how to do in just a second. There's also, of course, going to be differences in biodiversity among regions. So, for instance, you're going to have regions of Europe that have high biodiversity and regions of Europe that have lower biodiversity. And so if you, for example, are calculating changes in the raw values of your metrics through time, let's say richness, often those changes are not comparable among regions. For example, if you have quantified that, oh, this site only experienced a change of 2 species per year, whereas this site experienced a change of 10 species per year. Well, those changes aren't actually really comparable to each other because 2 species per year in, say, Northern Europe could actually be pretty high. And 10 species per year in Southern Europe could actually be kind of low. And it's simply because their species pools are totally different. You're also going to run into potentially identification differences among regions. We encounter this problem a lot with invertebrates. It may not be so much of a problem with other taxonomic groups like fish or plants, which tend to be identified always to the species level. But certainly with other taxonomic groups like invertebrates, you're going to end up with mixed resolution data. So for example, what you're looking at here is a list of taxa across these sites. And this is a different data set. So this is Sweden, this is Spain, this is a different data set. You have a different list of taxa. You can see that these ones have a mixture of like species and genus and family level identifications, whereas this data set is almost entirely identified to the family level or higher. This causes a difference in diversity between these data sets and even between regions that are not related to actual biodiversity differences. It's simply a difference in identification. So you again have to potentially control for this issue, which again, I'll mention in just a second. You're also, of course, going to have to harmonize your taxon names. What's a taxon that's called one thing in one region may be called something different in another. And so you need to make sure that everybody is using kind of the same names. And so we, for example, create sheets like this that list what is the every single unique taxon name that has been sent to us. And then over on the right, whether that taxon is to be kept in the data set or removed. And if it is to be kept, what is the actual name we're going to use for that taxon across all data sets. All right, lastly, you're of course going to encounter methodological differences among sites and among regions. I'm just highlighting a couple examples we've run into with the invertebrate data. Here we have differences in various methods across countries. You're, of course, going to potentially have differences in, let's say, abundance units. Some countries are going to record things in individuals, let's say, per sample or per square meter. You're also going to have differences in the time series themselves. Some are going to start and end in different years. Some are going to have a different total duration. And so this is also going to create some potential problems. All right, so given all of these issues that I've outlined, how do we actually go about, let's say you have a biodiversity data set, how do you go about quantifying how things have changed through time? So this is just a general guide. It's not necessarily going to apply to everybody, but it's, kind of a steps that we follow that have seemed to work pretty well. So first, of course, is to, if you want to calculate a slope, so you want to determine how has this site changed through time, choose your method for doing so. So the linear model is, of course, going to often be your method of choice, but some may want to get a little more complicated. Maybe you want to account for nonlinear dynamics through time, and that's usually fairly well dealt with by GAMs, so generalized additive models, but there's other options. You could go a Bayesian route to deal with it. And this GAM actually includes a first-order autoregressive structure, which deals with temporal autocorrelation. And that's been a pretty effective way we found of dealing with that problem. So it's often called just an AR1 term that's included in the model. But as you'll notice, the slopes are actually not really that different between these two methods. So you could go through all of these ways of controlling for issues, and really it kind of just ends up the same because, again, A basic linear model using the normal distribution is actually fairly powerful and robust to a variety of violations of assumptions anyway. Step 2, once you've dealt with, how do I actually want to extract my slope values, you need to deal with this issue of standardization, which is how do I make these slopes comparable among different sites in regions? A couple of the easiest ways we found of dealing with that is simply to standardize the data. So this is, for example, converting it to a z-score, which is standardizing it to the mean and standard deviation of, let's say, each region the data comes from. So what you're calculating is actually a change in standard deviations per year. And so that helps things out because, the mean of richness in, let's say, Sweden and how it changes in relation to that in terms of units of standard deviation can actually be compared to the mean of richness in, let's say, Spain and how a site changes in terms of that standard deviation. A more recent way we've dealt with this problem is instead of calculating just the regular slope value is extracted as a percent change per year instead, which just involves log transforming the original data, and then you extract the slope and exponentiate it and subtract 1, and that converts it to a percent change per year times 100, of course. All right. Once you've calculated, you've determined, okay, this is how I want to calculate my slopes and fix these issues of comparing among regions. You then repeat that process for every single site that you have data for. This is kind of what it looks like. So this is, for example, the slope value of richness, so how richness is changed per year in each site. Then you construct your model. So you've got your response variable, slopes in whatever, relate it to your predictor variable, and include some control variables to deal with some other problems. In our case, we're looking at slopes in biodiversity, relating it to slopes in ecological quality. For control variables, it's usually the same kind of set. You might be including something like latitude and longitude that deals with broad east-west and north-south differences that might be arising in your data. For spatial autocorrelation, we found something like Moran's eigenvector maps can be quite effective as just including as predictors that map out essentially spatial autocorrelation among your sites. You could include a control variable for the length of each time series. So that controls for differences in your slopes that may be coming from the fact that you have a shorter time series in some sites and a longer time series in others. And you usually want to include, let's say, a random intercept for the country or the dataset provider. And that controls for just a variety of things that may differ for various reasons among countries, like methods and so on. All right, so in the last minute, I'm just going to show you some of the results that we find out real quickly, and then I'll finish up. So here we're looking at the y-axis. These are the results from our actual analyses. The y-axis is the slope and richness, so how richness changed through time, and the slope in the EQR, how it changed through time. These sites, the values on the right, EQR is increasing, so things are improving in terms of getting closer to reference conditions. On the left, things are getting worse. What we found in terms of richness is that actually you do tend to see somewhat of a consistent positive relationship between richness and ecological quality. As ecological quality increases, richness also tends to increase. But of course, you see also a fair amount of variation. There's a lot of sites, let's say in these white squares here, that show the opposite response. Now, when we look at other biodiversity metrics, let's say abundance, we found a much weaker relationship. So as ecological quality changes, there's no consistent change in abundance. It can increase, it could decrease, it could have no change whatsoever. There's no real indication that you can use abundance to say anything about how impacts are changing. So we'll finish up. Can we infer anthropogenic impacts using biodiversity trends? Our results basically said for some metrics, yeah, probably you can. There's still going to be a wide variation in responses, so you need a large data set to kind of pick out these overall responses, but it can still be pretty effective. However, for other metrics, no, they don't tell you anything about what's really going on when aggregated across all of these different communities. It may be informative for an individual community, but not necessarily across a broad scale. And I just wanted to highlight in this last second that the questions that we are dealing with and the types of analysis we were talking about can be done entirely just using community data. So we did not need matching environmental data. And so there are a whole lot of questions out there that you can answer, including using the eLTER data just with community data. You don't necessarily need to feel like you have to go out and find all of this important environmental data and deal with it. All right. The data I've shown you is now publicly available if you want it. So the ecological quality data is in this publication right here. And the community data, including that which Peter talked about, is in this publication right here. So it's all openly accessible. All right, that's everything for me. Thank you very much. And Peter and I are happy to take any questions that you have.
Thank James and thank Peter for your presentation. Actually, very informative, detailed, and also considering a lot of methods and approaches. So I opened the question and answer session. Actually, we had a question on abundance that was answered in the chat. So Thomas, a question from Thomas.
Yes, please. So really cool stuff. So thanks a lot. Exciting data set. I just have one question regarding one concern, let's say, regarding this reference you use for defining the ecological quality. Can you go a bit more in detail there? I mean, this is of course criticizable. Who is defining the reference, at which timing point? What's the context there? It's always kind of a value proposition behind that. So I mean, just reflecting a bit also from a policy perspective, like for the Habitats Directive, this concept is used a lot and criticized a lot. So maybe can you give more context on that?
You specifically mentioned Peter, so I'm going to leave this to him, but unless he wants me to deal with it.
Yeah, I think it's going into a similar direction to the question that Luciano is mentioning in the chat. Yeah, so maybe you could briefly provide some background information with regard to the ecological quality and how it is calculated.
Sure. So we've also, in publishing these papers, we've run into similar questions about how useful are these ecological quality values really, because they're calculated in different ways in different places. They use different reference ecosystems. They can be tuned to detect different stressors. And so can you really take this data and, essentially compare it across Europe? We've outlined, including in these publications, ways in which they are not useful and ways in which they are for our particular question. So the way in which they're not useful is they tend to not tell us anything about particular stressors. They can be tuned to detect particular stressors, like say, you could present an argument that ecological quality in some countries has been specifically parameterized with, it's basically comparing parts of the community that are supposed to respond to organic pollution to those same parts of the community in the reference ecosystems. But what we find is that across Europe, the same types of taxa tend to respond to a variety of different stressors, as are the same taxa that respond to organic pollution. So you do have general patterns across Europe in the types of taxa you tend to find in damaged ecosystems versus the types of taxa you tend to find in more pristine ecosystems. For example, in the study I presented, we also looked at EPT relationships to ecological quality, and you find those relationships happening across Europe. So yes, you can't use the ecological quality ratio or ecological quality classes for certain things, but if all you're trying to do is say, in these communities that tend to characterize damaged ecosystems across Europe, do they tend to be lower diversity communities? In these communities that tend to characterize less damaged ecosystems across Europe, do they tend to be higher diversity communities? Those questions you can use something like the ecological quality to answer.
Thanks.
Thanks, James, for the answer. Is there any question? Actually, you see in the chat that the the material is also provided there. So the links to the papers and the presented studies. I want to mention what Ulf wrote, that the data calls that were mentioned by Peter then were the basis to create the eLTER marketplace, where that is a place that you can use for actually publish your call or and also contribute to other calls. And exactly, it's more than 100 sites and platform coordinators that are there. So this is also a tool that actually came after the successful data calls that were presented by Peter. If there is no any other question, we will move to the presentation by Christian Poppe, as we have time before lunch, and actually to go through the next presentation and study on linking modeling and observation by Christian Poppe. So I leave the floor to you.
Okay, thank you. And hi everyone. I'll share my screen now. Okay, this seems to be working.
Yes,
yeah, just
perfect.
say something.
Perfect.
Good. And you can also hear me, so we can just start. My name is Christian Poppe, and I'm working as a postdoc in the Research Center Jülich in Germany. And since my PhD, I've been working on a pan-European ecosystem model, but it's always connected to the eLTER network and to station data. So this will be mostly about just connecting the data in observations from the eLTER network as a whole, and also sister or co-located networks with this pan-European large-scale ecosystem model. And I've done this together with these collaborators that are listed here in the Research Center Jülich. Okay. Don't know. Am I still sharing the screen? No.
It was black.
Yeah, sorry for that. I'm not sure why, but it will be up again in a second. Let's do it like this. Okay. I don't know why, but it's not.
Go
not...
over the presentation and just over there should move.
Yeah, just a second. I think something was wrong with the setup because I was not able to change the slides. Let's try again. Okay. This is fine now, right? Okay, I assume
it's moving.
Okay, perfect. Yeah, so why we are doing this type of research is because we're interested in this large-scale approach and local differences across the large-scale continental scale of the different ecosystems. For example, how they respond to climate change and the increasing drought severity that is projected across Europe, also in different ways. And we're specifically interested how carbon fluxes and water fluxes, for example, the carbon uptake from the ecosystems from the atmosphere, because it would then also affect the carbon balances again in the atmosphere. So there is a lot of feedbacks, and of course that depends on the different ecosystem and the climatic conditions that they are in and the changes in them that they are then susceptible to. And we're looking at the pan-European scale, and for that, in the beginning of this eLTER Plus project, we have conducted a small survey that we send around to the stations, and we basically ask where and what variables that we need are being measured. And on the left-hand side, you can see the sites that basically have any type of hydrological variables available. And there are quite many, but there's also some gaps or disadvantages here that we have to consider if we want to do large-scale analysis. So first we have this sparse distribution geographically of these data points if we really want to have like an unbiased, like consistent distribution across the continent. And then, of course, we have also an inconsistent length of the observation time series. So while some sites have long time series for a certain variable and others would have maybe only a year or less in some cases. And then also depending on the variable. So we might have a very large set of variables in some of the more equipped sites, and some other sites might only offer a subset of the variables that we need to do a very comprehensive hydrological analysis. So that's where now the contrast to the European land surface model comes in. And specifically, this is the community land model versus version 5 in our case. And we set this up in 3-kilometer spatial resolution for a simulation period from 1995 to 2018. And there we have 16 different plant functional types. And in contrast to a site network that has a geographical bias and so on that was already described by the studies of Thomas, here we have a very consistent spacing of the grid cells. So we have the 3-kilometer everywhere and also no gaps in the time series, of course, and also the same set of variables, which is actually a lot of variables. And we're able to do some more comprehensive analysis. But of course, we have to say here that we shouldn't take the model as the right or the truth. Of course, the site observations are the gold standard for the ground truth of the data and the variables. But if we evaluate the model and integrate these two data sources, we are able to get, hopefully, and to the best of our understanding and knowledge, the best of both worlds. So a robust data set that has the spatial and temporal gap-free time series for a lot of variables. This is another example of the ICOS research infrastructure network. These are 72 sites, and this ICOS format or this ICOS published data sets, they offer a harmonized format of all the variables that we basically want to look at hydrology and carbon cycle. Specifically for the carbon cycle, I think ICOS is the gold standard for with the eddy covariance stations and the half-hourly measurements of the carbon and water fluxes. But again, there are some biased geographical coverage, as with any in-situ observation network. But it's also very co-located with the eLTER sites and facilitates then the whole systems research. So we will use this network also to compare and evaluate the model and do some further analysis with it. And now to some more technical issue that we encounter when comparing the site data with the data from the European land surface model. So as I described before, the model has this equally spaced grid cells. And you can hopefully see here the equally spaced grid cells and the grid cell centers in black. That's basically the location of one grid cell. And then the grid cell borders, so just the division between the grid cell, the area represented by that grid cell, is that also in black, the dashed lines. Then we have basically a station and that red dot that is located somewhere that, yeah, in our model setup, we do not specifically take care of aligning the grid cells with some sites. So we have this predefined domains over Europe that many people have done a lot of effort to basically define the extent of the domain and the exact amount of grid cells. We have this equal spacing and the representation of the region, also compatible with some global approaches. So here in this, when you also look at the footprint, so this the area in the dashed lines and within the red dashed lines of the station, you can see that we have some type of dilemma. So this, let's say this is an eddy covariance station, and the footprint basically measures fluxes from, let's say, the forest, let's say this broadleaf evergreen, broadleaf deciduous forest, let's say, and we want to compare this to some model output from this continuous European land surface model. And we most likely would choose the approach to compare it to the most near or the nearest grid cell. But the problem here is that the grid cell center, that is here the right in the middle one, if you can see my mouse, would also represent or mostly represent agricultural area, even though a part of the grid cell is still the forest that is measured in the observation station. So there's some ways to deal with this, and the best way, for example, you could just look at the surrounding grid cells, and maybe one of the lower grid cells here has a larger coverage of this forest, and it may be a better comparison to the station if you actually want to evaluate how your model is and how well your model is simulating exactly this forest. There's also other methods to basically find the grid cell that's most close to the characteristics of the site, and then the best approach to compare it basically. But in the newest or latest generation model, we can also look at inside of the grid cell basically and extract the time series in the closest grid cell. So again, this on the right, that corresponds only to the forest. So we would have the closest matching also functionally, because we are not comparing the aggregated grid cell that is somehow basically representing a weighted average of the all the land covers that are in the grid cell. So here that's both crops and forest, but we could only look at the closest grid cell and only look at the forest that is within that. And in the community land model version 5, this is done with this tiling inside of the grid cell, where first we have the land unit, then which is like the vegetation, then a possible lake. So there's all inside of one grid cell, urban tiles, crop tiles, and then the vegetation is basically further subdivided into different soil columns where you could have competition for water. And then finally into this plant functional type patches which are the actual functional representation of the vegetation in such large scale models. And these are, for example, different types of forest, and there the distinction is between needle-leaf evergreen trees and broadleaf deciduous trees, and so on, different crop types, grasslands, and so on. And then for the forests and grasslands and shrublands, at least there is this further bioclimatic division between the temperate deciduous broadleaf forest and the tropical deciduous broadleaf forest, and so on. And then within the grid cell, and for each plant functional types, then the model calculates basically all these equations for the energy fluxes, the hydrology. So we have the energy cycle, the water cycle, and the carbon cycle represented and interconnected with each other, because we have this biogeochemical cycle module also turned on. So that means that the carbon taken up, so the photosynthesis basically depends on the climate and the water, and also the nitrogen. So the nutrients also play a role. And then from this carbon taken up, we can also then further calculate the leaf area index. So it's not like calculating the leaf area or the carbon taken up from leaf area that we prescribe from remote sensing, as it was usually the case in the previous generation land surface models. But now we're able to fully resolve this carbon cycle and actually have this dynamic leaf area index depending on the climate, and also compare basically this carbon model by comparing the LAI, for example, from the model with the observed time series. So this is just then for your information, basically, with what type of plant functional types are represented across Europe. And this is just a dominant PFT. So as I pointed out before, one grid cell has most of the times different plant functional types present. But this is the dominant, and you can see that this purple is basically the unirrigated crop. That is the dominant PFT across most of Europe. And then in the north, we have this light purple. Yeah, I don't have a better description for this color. And the dark blue, so you can see this the transition from the needle-leaf evergreen tree boreal to the needle-leaf evergreen tree temperate. So that's like the bioclimatic order that distinguishes these two plant functional types. And now the question is still, if we want to compare this to the station data, we want to closely look at how the grid cells then represent this. And this is an example for an ICO station in Tarent in eastern Germany, close to Dresden, where the station is representing the evergreen needle-leaf forest. As you can see from the satellite picture, also in the cross is the exact location of these of this station, and the circle is basically the 1 square kilometer footprint area. And then this whole picture that I'm showing here would actually, with the black cross in the middle, is the grid cell center of the closest grid cell. And the whole satellite image is basically the grid cell of the 3 times 3 kilometers. So we're able to see in terms of land units, we have more than 50% natural land unit, and less, basically, than half, let's say 40% crop. And that basically compares well to just by vision to the satellite image. And now looking at the plant functional types, that is the pie chart on the right. That is within the natural land unit, so only looking at the green part of the pie chart on the left. We can see that most of it is actually saying it's a grassland. So I would say this doesn't really fit well here, even though the evergreen needleaf forest is still represented. So this is there's still some bias here that might arise just from the remote sensing data, land cover data that is basically derived from remote sensing that is then used to create the input to CLM5 about the land surface information. But as I said, still we inside of the grid cell, we still have this evergreen needleaf forest. And if we are able to or extract the time series for the needleaf evergreen forest in this grid cell, we can have a very close match, hopefully at least, of the functionality and the represented ecosystem. So now we're going to look at some evaluations. So this is for the same site, just the time series. So here for ICOS and Talent, we have the pretty long time series. That's why I chose this example from 1996. And here we compare to the grid cell SELM5 values, so not the PFT level, and some remote sensing. So we're able to see the seasonal variations are very well covered, even though both of those data sets slightly underestimate, or sometimes not so slightly underestimate, the summer gross primary production that is measured by ICOS. And sometimes there's also differences between the remote sensing and the CLM5. For example, in 2005 in summer, we somehow see a reduction or reduced grass photosynthesis basically in CLM5 that is not seen by remote sensing or the ICOS measurements. So that would be also interesting to see why this exactly this year and so on. But because we have this large scale analysis, basically this is extracted from this large European model, we are usually not looking at specific years and specific sites, but basically aggregating over the whole domain to get a more large scale meaning out of this. More on that later, but first some more examples about the validation of, for example, this is remote sensing. As this is also remote sensing has a large coverage. We're able to look, for example, here at the R² of the glass, that is a remote sensing product, ET, evapotranspiration, compared to FluxNet sites, and see mostly good R² values, but at some sites also low R² values. That might be because there's also maybe also a mismatch in the represented ecosystems or short time coverage. And basically, this is another example of this type of evaluations across the continent, where we're using also different data sets. So this is the FluxNet and the ICOS for both the GPP and the ET, for still evaluating the remote sensing data sets of GPP and ET. And basically the R² values show good agreement, but then again, in the agreement, we sometimes see some geographical bias where Central Europe is very well represented, it seems, but Southern Europe less so. So maybe that's also related to how more arid ecosystems are represented in this remote sensing models to basically translate these images to these ecosystem processes. And then we have a less good match for the subflow, that is the map on the right, SubFluxNet is basically a large-scale data set for slab flow measurements, and subflow measurements are basically on trees, but here we can also then upscale these measurements if they are you have multiple measurements in one forest and they represent basically more than 80 or 90% of the present tree species. We're able to upscale based on the basal area of these trees and then basically compare it to the forest that we simulate in the model. But still, we here the comparisons are not so well. So we expect here some scale differences responsible for that, even though we have we discovered a similar magnitude of the transpiration here. The variation is not well represented between these. And this is now a more sophisticated approach to evaluate the model. So here we, as I hinted before, we aggregate the sites across Europe to basically the plant functional types. So this is a Taylor diagram that basically shows 3 different indices of evaluation. So this is the correlation here as the angular index, basically the standard deviation here on the X and Y axis. And then inside you can see this gray circles that are basically mapping the distance from the reference dot. So that is the black dot where the black dashed line ends on the x-axis. That is the reference measurements. And for example, here in the upper right, that's for the evergreen needly forest, we see in colors basically the different data sets. So here we have the remote sensing in green and some reanalysis that fuse basically the models with observations in brown and in purple. And then our land surface model in these 2 levels. So the grid level, so where we have the grid cell time series in red, and then the yellow where we have the PFT specific. So in the case of the evergreen needly forest, for example, we would only look at the evergreen needly forest of the grid cell in this yellow markers. And what we discover here basically is that we have always like a very similar level of correlation for all the represented data sets compared to the ICOS observations. And we'd also discover that it is basically sometimes not given that the PFT level representation or scale inside of the CLM model is better than the grid cell level aggregates. And yeah, this could be also for many reasons, but basically the interpretation here is that the functionality of these plant functional types inside of the model is not representing what really goes on, or like the variations in of the ecosystems are not well represented of this PFD level disaggregation of vegetation inside of the model. So there is a lot of discussion about this, and maybe you'll also have questions. Happy to discuss about this actually. Yeah, now going to more after having evaluated this, going to this more advanced variable that we analyzed, which is the water use efficiency that basically combines this carbon and water fluxes. Basically, it relates the amount of carbon taken up by the amount of water that is evaporated. So how much water does an ecosystem use to assimilate a given amount of carbon? and we calculate then the trends over Europe. And we have these 3 maps, and then for the stations, you would see mostly red colors. There's our negative water use efficiency trends. So that means that across the study period, the ecosystems were actually using more water to assimilate a similar amount of carbon. And we see this same trends over Central Europe that we discovered for the stations, for the remote sensing, and a little bit more differentiated inside of the CLM5. But as another example, or another note, is that for the large scale data sets, the remote sensing and the CLM5 were able actually to see large areas in Eastern Europe with large decreases in water use efficiency that we are not able to validate with station observations because of the missing stations there. And basically also the trends in Northern and Southern Europe are less well validated, or their significance is not very represented or present in the station analysis as compared to what we see in our large-scale data sets. We also looked at how this water use efficiency responds to drought, and this is a more qualitative diagram, but there's also the quantitative, so with actual numbers in the study here, that will, I think, also be linked in the chat soon. But basically we see that the water use efficiency responds negatively to droughts or dry conditions, and it somehow also shows a gradient across the hydroclimates. So here we define hydroclimates based on annual precipitation. That's on here on the right-hand side, you can see the map, how the hydroclimates here are distributed across Europe, and we see the strongest response, at least for the dry conditions, for the most dry conditions for semi-humid hydroclimates, and that's a negative response, while for wet conditions we see a positive response. And we were also wondering how or why and what are the drivers actually locally for changes in the water use efficiency. So here we also did a large-scale analysis using this causal network approach, this PCMCI. And for every grid cell, basically, we came from this initial network where we just had some assumption how the variables are related. That is this network on the left-hand side. So there you cannot see any color or something, just the connections that are basically physically possible. There, arguably there's more or less connections, but it really depends on the local scale and how these connections basically emerge based on the local characteristics and basically also the climates. And on the right-hand side, basically, this is then the output of such an analysis for one location. So you can see that now some of the arrows basically vanished because there was, for this location at least, no significant link discovered. And the other links are basically coded by how strong and also which direction, so positive or negative, in which direction the variable influenced the other. And here we're looking at basically the water use efficiency that is basically related to all the variables here indirectly, but of course directly the changes are caused by either the gross primary production increasing or decreasing, or the evapotranspiration increasing or decreasing, or both of them increasing or decreasing, or none of them. yeah, so we're interested how this basically comes out at the European scale. So this was an example for the local scale. So you can imagine if we have one graph for the whole, for all the grid cells, which are a lot, we have to somehow then abstract this. And in this example here now, we're looking only at these two links. So the link from gross primary production to the water use efficiency, and from evapotranspiration to the water use efficiency. And in this map on the left, you can see that where it gets more green, basically that means that gross primary production was causing more of the variability of the water use efficiency, and where it's more pink, that is more evapotranspiration related variability of the water use efficiency. And this mixed colors in the middle towards the dark blue, basically both are roundabout causing the same amount of variability and in water use efficiency. And you can see again here similar patterns to the hydroclimates here on the right. So our assumption or our interpretation basically also in the paper here, where we go into this in more detail, is that in more arid ecosystems, we see that gross primary production variability actually is governing a lot of this water use efficiency variability, and basically the evapotranspiration is decoupled from this. Yeah, this was a lot of analysis, and maybe you're also interested to do some similar stuff, or just have a look at how a data set from this large-scale European model actually looks like. And recently we had published also in this eLTER Plus project context a data set that includes the simulated variables across Europe, and you have everything you need there to actually reconstruct these analyses. So all the variables and across Europe, and you have all the years. So here's the QR code and the link if you want to have a look. Basically, that's also a possibility to have some hands-on or just let's do some data discovery. Yeah, so I think if you follow this link, you have to do some click. There's some link for the data access, and then you will end up in this index website where you can see the yearly netcdf files, and basically you can download them and open them on your local computer, but 1 year is basically 36 gigabyte. So if you want to have a look, maybe focus on a few years only, if you are looking at it on your local computer, or it's also possible, very easy to download this on a cluster computer, basically to do some more long-term analysis. Yeah, but yeah, it's just some technical recommendations if you want to do similar analysis. Basically, here's the land surface model that we use is the ECLM. That is basically the ECLM5 with some additional developments from our group. And for the European 3 kilometer setup in high performance computer is basically recommended. So here for this model, we used over 1,000 processors to simulate 1 year in a little bit over 6 hours. And if you're interested in the exact setup of this domain, you can also have a look at this GitHub page and some technical recommendations also for the visualization of this data set that I mentioned. So here you have netcdf variables, libraries, and NCView. That is for this quick visualization, and basically everything I have showed you today. I did it in Python, And these are just the very useful libraries that I use in Python. And DIMES here is very good actually to look at the station metadata. So here you can compare or extract basically data for each site. So that is related to hydroclimate already. So basically the mean annual precipitation or just the location coordinates, for example. So that is very useful to do. So you don't have to copy paste this from other Excel files or the internet websites of the sites. Basically, you can do this systematically with this library. Okay, just now to summarize this, I hope you took away that it's very important to quantify these variabilities of the ecosystem processes, as they're also then later linked to the fitness of the ecosystems, and that different data sources have different qualities, but all can be used and if used correctly, they're useful, and that we follow an integrated approach, and that also we are continuously integrating development in this model. So we also have some newer outputs where we have a more novel input data regarding, for example, this PFD distribution across Europe, and include some further PFPs. So for example, apple trees are now represented that were not in there before, and that we're also looking to integrate more multiple models and have a more transdisciplinary approach here in future, basically. But there's also the possibility to more robustly or have this data assimilation approach, where we directly fuse the observations and the models and basically create a very comprehensive data set over Europe that is basically also statistically considering the errors of the model and both the model and the observation, and basically have the best estimate while still having this gap-free time series over Europe. We're working also towards this, to have this ecosystem reanalysis, where we also integrate the ecosystem observations into the model and update parameters correspondingly. Okay, that's it from my side. Feel free to ask any questions, and also after the workshop, contact me via email, for example. Thank you.